Well, good afternoon. We recognize we're the last session of the day, but it's an exciting topic, and we really look forward to sharing perspectives with all of you. Again, my name is Dr. Justin Gaynor. I'm a thoracic medical oncologist. I think John's been up here a number of times today. So maybe we'll turn to our panelists and let each introduce themselves.
Good afternoon. My name is Moitrey Chatterjee Kishore, and I'm the head of immuno-oncology and cancer cell therapy development at Astellas. Nice to be here.
Yeah, hi, my name's Neil Martin. I'm the CEO of a biotech company in the UK called Artius, and we specialize in the area of DNA damage response.
And hello, my name is Chris Varma, and I'm the co-founder and CEO of Frontier Medicines.
Great. So our topic today, we're going to be covering a lot of ground, ranging from precision medicine to antibody-drug conjugates, bispecifics, T-cell engagers. But I'll start with precision medicine. So Chris, we're about 20 years in now, since the discovery of EGFR mutations in lung cancer and imatinib and CML. How do you think drug discovery has changed now for the next two generations of precision medicine?
Yeah, no, it's a great question, Justin, and honestly a question that I could talk about for an hour, but I know we don't have that kind of time. So I think, just a few initial thoughts. I mean, first off, what, drugs like Gleevec have done for CML patients, right, or, drugs for EGFR mutations is really phenomenal in terms of response rates, in terms of durability. And, but there was a whole history there, right? And so I think one of the things that those targets and the work done on those targets have taught us is if you look at, for example, EGFR as, first generation molecules such as Iressa, essentially delivered kind of a, 40%, 40% response rate, 6 months of durability, and there wasn't much kind of patient uptake until you really got to next generation molecules like Tagrisso, where you had 70% response rates, 18 months of durability. So really providing true value for patients and for the clinicians that treat them, and that is, I think, one of the most important lessons. And what's, I think, happened now is essentially where we've gone from kinases like EGFR or ABL to KRAS, P53, really truly undruggable, one would say, holy grail targets of cancer, and applying the same types of methodologies. And so I think that is one of the big changes, and we're obviously, cracking targets like KRAS. In fact, that's something that Frontier is working on. And we found that there are different approaches to address resistance, in that case, particularly on and off. I think the other is really around just the paradigm of treatment. And I would say that while there is, I think, a lot of optimism in terms of what we can do now with precision medicine, I think there's challenges that remain. And frankly, those are really having to do with essentially the clinical paradigm. So, the treatment of or your identification of patients, right, is really in a construct of the organ, lung, colon, et cetera, right? And for a really good reason. But you often don't have the genetic profiling that goes along with it. At a great place, here with Mass Gen Brigham, most of those patients that come in, right, are going to be in some sort of way sequenced or profiled. That doesn't happen in most non-academic medical centers, and so that is a challenge that I think still remains.
Yeah, I think that's a great point. I can say even in the clinic, right, when we talk about the easiest biomarkers, right, very simple things like KRAS mutation, uptake's not 100%. So then as we start thinking about even harder biomarkers, that is going to be a challenge moving forward. So maybe a springboard, you mentioned, we've been focusing on, the last 2 decades have been focused on kinases. And you named a couple non-kinase targets, but maybe opening it to the broader panel of how do you think about, what are the exciting targets in terms of precision medicine?
I think, obviously in my space, which is in the DNA damage response, You know, the targets, the enzymes that we're actually looking at are getting tougher to actually drug in many respects. The low-hanging fruit, such as kinase and PARP inhibitors, you know, were really well identified and drugged, but certainly over the last sort of 5 to 10 years, we're now having to focus really in on the more specialist enzymes, you know, polymerases, helicases, nucleases, and therefore I suppose the methodology of how we drug them has had to change. But I think that's fine. I mean, I think that the technologies are out there now to actually do that, and that's why I see a real pace change in how we can actually drug these targets. You know, PROTRACs, you know, DNA, you know, chemistry libraries, all of these things, you know, good co-crystallization, structural work really helps us identify these enzymes and actually drug them, and this is where we're actually now starting to see that benefit. So we actually are getting there, ultimately, with these new targets. And I think certainly in terms of the protein-protein interactions, that was a big area that was really tough to actually identify good inhibitors. And now, you know, as I say, with things like the degraders, good peptide biochemistry, chemistry, it is really making a big difference to us actually working on tough targets like P53, for example.
Yeah, I think that's a great point in terms of just disrupting PPIs. The approach that we use is covalent drugs and approaches, and in fact, those are very efficient mechanisms to disrupt those types of targets, which again we've learned right over the decades.
Dr. Chatterjee.
Yeah, I think we stand on the shoulder of giants. The amount of work that has been done and supported by, you know, people who are represented in this room, investors, stakeholders in regulatory sciences, of course, developers, investigators, has led us here. And at Astellas, I think for us, the important thing is doing the things that we have the best capabilities in and supporting patients throughout their journey with cancer. So we are really focused on GU cancers, GI cancers, where we have established certain drugs using different modalities that we have developed. So in the last 3 to 5 years, we have spent a lot of time developing a toolbox of modalities and using the scientific advancements in partnership with organizations and institutions like Mass General. We are trying to dive deep into the disease biology. What happens in the first line of treatment? What happens after that? How does the first line of therapy change the disease itself? The tumor itself adapts and resists. How do we attack that? So because we have the whole toolbox of modalities, we're now beginning to pair up targets with the right modalities for the right patients. We are really patient focused. We are thinking of what does the patient really need at those different stages of disease, and our toolbox of modalities plus our disease knowledge is helping us do that.
Yeah, as I sit here today, I mean, one commonality between Astellas and Frontier, certain targets are in common, right, with KRAS being one, but, you know, we've seen KRAS being approached 1 million times before. You know, the safety profile is not always great. We've seen other people try and fail completely, right? I guess, you know, Chris, starting with you, and then we'll move over to Astellas, but how has your approach kind of elucidated KRAS as a target for you?
Yeah, no, thanks for that, and you know, completely agree with you that, you know, certainly while there has been success in terms of actually drugging KRAS, like 12C and other mutations, there is still a tremendous amount that we need to do to really further patient care in that space. And so Frontier's approach really has been to utilize covalency and covalent small molecule drug discovery with a technology called chemoproteomics or chemical proteomics, which essentially allows us to not only kind of drug any target that we want, but to do it in a very, very selective manner. And that's one of the key points because, you know, what you need are extremely clean molecules that ultimately can be combined. And we know this from the antiviral world. I think the oncology paradigm is actually very similar to that, where ultimately you need to be able to address a tumor from multiple different points of nexus in order to completely shut down signaling. And so what we've done at Frontier is, as we develop all of our programs, including FMC 376, which is in the clinic, which is an on and off KRAS inhibitor. So one, completely shut down all signaling through that pathway, but two, to do it extremely selectively, utilizing proteomics at every stage. So we know every single off target in the cell to our molecules. And of course, we strive to deliver extremely selective molecules so that we can combine them ultimately.
And how has Astellas approached these kind of small targets, small molecule targets like KRAS?
Yeah, so we have our targeted protein degradation group that is working on several different E3 ligases. But I think the quest that has taken us to ADC approaches or RLT approaches has been how can we do a precision-guided bomb, in a sense, to the tumor. And approaches like you are applying. For us, it is really marrying the technologies that we have, from antibodies that are super specific to the particular target and attaching different types of payloads to them, whether that is a chemo, whether that is a protein degrader. So we are trying to make these therapies more and more precise. Also, the potency of these molecules is important for killing, but to manage the potency in such a way that it doesn't lead to off-tumor activities, which have limited the use of ADCs beyond a limited amount of dosing cycles. And so if your patients don't get exposed to the drug, they don't see the full benefit of the drugs. Therefore, a lot of these drugs are being challenged by the lack of overall survival data in this modality. So what we are trying to do also is to make these molecules potent and also add in moieties such as immune targets, so that we have a partnership with Sutro where we are developing immune ADCs. These molecules are potentially even more potent, but they can be dosed in a fashion where they can be effective at much lower doses that will allow for activity in monotherapy, but also allow them to be combined with standard of care, which is also important in different lines of therapy. So our toolbox allows us to mix and match. It is truly target and disease state dependent what we match to each other.
I mean, this whole question of combination, I think, is really important because I think that where we were before with these untargeted agents, it was very difficult to marry up mechanisms. But now with these more targeted agents, and we see this in the DNA repair field, for example, is that you can start to marry them up and you can get more effective effects at the tumor. And it's building those layers of damage, of inhibition that ultimately can be far greater in its effect at the tumor. If you can make that more tumor specific rather than, and I think that this is the whole, for me, this is the exciting future of oncology is actually being able now to really mix and match different modalities because we struggled before because of the systemic effect of them. And that for me is really important.
And take for example bispecifics. I mean, I started in the field maybe 10 years ago in hematological malignancies, where it was really bringing the T cells to the tumor. It is a simpler approach, dare I say, to my hematological oncology colleagues, than in solid tumors, because the target is much more proximal. So you bring the T cells to the target and you see the killing, and it's simpler to actually visualize it. In solid tumors, you actually need to match two different mechanisms. And so we are now seeing the evolution, which we have been trying to do for a long time. We have the EGFR MET, but now with the VEGF PD-L1, we are seeing how we can not just bring T cells, but bring two different targets together so that the 2 + 2 equals more than 4, hopefully, as you pointed out. And I think the design of these molecules is going to be critical.
Yes.
But also innovation lies in making these agents more accessible, easier to use, hopefully more tolerable, so that we
Yes.
can allow our investigators to use them for a longer duration period.
think that's a really interesting point. And as we start thinking about the mixing and matching, how do you start then deconvoluting mechanism of action? Like what is actually doing the heavy lifting in the tumor?
Right. And that's a really important question. I myself worked on amivantamab for a little while, but it was that question that we got: is it EGFR plus MET, or is it just being driven by EGFR? I think that question will have to be defined. Also, for in clinical dosing, but you are kind of stuck with one molecule that, if two different molecules targeting the two different targets, sometimes easier to titrate. And you have to really figure that out, perhaps using preclinical models, but also in the clinic to really design the trials differently. And we have been thinking of doing smaller randomized trials, for example, earlier on to really try to distinguish, is it really the contribution of the elements? And the FDA will be asking for that as well as we go on further. So the sooner we get the answers, the better.
I think, though, Justin, on top of that is that a lot of the modalities have been known for quite some time. It's just been impractical to actually do them. And in my field, you can take an example of the DNA repair and ionizing radiation. I mean, it's super efficient. It's super, synergistic to knock out your DNA repair factors, which are the resistant factors to ionizing radiation. But now with the RLTs, because they're so much more focused in the tumor, that combination just makes sense. And you're not getting that collateral damage that you were getting with ionizing radiation. So I think those modalities have been there. It's just been impractical to use them in some respects.
Yeah, and I think that from a discovery standpoint, Justin, I think it's really interesting that with the advancement of technologies now, like the ones that we deploy at Frontier, the question isn't even like from a phenotypic screen, what protein is causing a particular effect. We actually can narrow down to which residues of which proteins, right?
Yeah.
What sub areas of a protein. And that gives you exquisite ability to actually tune the system and both learn, understand, but also then go back in and tune it. So I think that and to do it at scale allows you really the ability to go after many different diseases in very different ways.
And if I may make another point, I had the good fortune to work on one of the first, I think, protein-protein interaction inhibitors, venetoclax, which is very interesting because that molecule, when we first started working with it, was a hard thing to formulate.
Yeah.
And a lot of innovations had to be put in place to make that into an orally administratable molecule. I think there is great opportunity for innovation in that field for protein degraders, for example. I mean, yes, reducing the size of the molecules, but to get it into a formulation that provides ease of use, that also hopefully reduces the tolerability package. Those will be important innovations in the next few years.
That's a great point, Maitri. And also, if you think about it, right, as an industry, we've been talking about 2 E3 ligases of 600 that are available that you can co-opt for targeted protein degradation. So one thing that we're doing at Frontier is we're going after many of the other ones. And what's really interesting is that they're actually specific in their tissue expression. So you can actually have built-in selectivity. So if you have, like, let's say a molecule that's sitting on the shelf or whatever in pharma company, or biotech company that, let's say, has a cardiotox, there are actually E3s that you can utilize to avoid any cardiac tissue and get the intended effect that you're looking for therapeutically. So I think that world is extremely powerful and rich to harness further for patients.
Now, we've talked a lot about matching pockets and residues and then the actual precision aspect, but then there's the clinical trial design as well, right? And harmonizing that with your product, enriching your patient population, maybe using a biomarker strategy, whatever it might be. And how have you guys approached it with maybe kind of like the clinical trial design to really perfect it to match the traits of your product?
Well, in my field, I think the big example was PARP inhibitors with BRCA1 and BRCA2,
Right.
that concept of synthetic lethality. That's a sort of power if you can get the biomarker right with the actual target. And you know, obviously we want to build on that and create more of those styles of medicine that actually do have a component, a tumor component, a genetic component, as BRCA1 and BRCA2 loss is, that you can actually identify quite easily in the patient's tumors, and actually then go for them. I mean, we're really interested in an area called replication stress, and if you think about all tumors, they're going through replication and dividing, and so if you can understand replication stress and actually target it in high replicating tumors, then you've got a real powerful mechanism there of actually stopping those replication, killing those tumors, and so you know, these are biomarker-driven events that you can pick up and utilize and actually build into strategy. So there's a lot out there, and the biology of the tumor itself, I think, is something that you can utilize to really understand how to target it. You know, mutations in KRAS, you know, DDR, you know, defects. These are all sort of big biomarkers that we can actually really target and utilize.
For us, it is all about understanding the benefit risk. I mean, obviously, these are agents that we are trying to innovate in terms of design to make them less toxic. But as we do that, we also have to understand who are these patients who will benefit the most from these. And so we are very engaged in understanding what markers we can use to stratify the trials early on and then select, hopefully, the patients who would most benefit, for whom that benefit risk makes sense.
Yeah.
Yeah, I mean, I guess, John, you know, I'm not going to lie. Like, the clinic is, you know, expensive and challenging. And, you know, I used to tell my team, you know, time to patient. And, you know, that meant, okay, you know, from our platform, we develop a molecule and we get into the clinic for patients. Now, like, time to patient to me means, like, getting it, you know, getting a molecule out there through the clinic, of course, vetted, and then to patients in, you know, worldwide, you know, broadly who need it. And it is, you know, while we've been able to make discovery very efficient, the clinic is definitely, you know, a challenge. And even for you know, approaches that we take, precision medicines, I mean, there is no lack of patients that have KRAS G12C mutations. Like, you know, absolutely. But there's, you know, there is a paradigm here for a good reason that, you know, does take time. And so, you know, it is challenging when you're in there, and, you know, every day you're talking about, okay, you're in dose escalation, there's a patient, you know, you hear about their story and you know, and the burden that, you know, they carry and the molecule that you have that can really help them, but you know, you have to take your time, obviously, right? And so I think that remains kind of a challenge.
But I also want to take it back to what you said, Justin, about even at an institution like MGB, right, getting biomarker testing is tough. Think of it in the community. I mean, how do we inform patients, how do we inform physicians that there are these markers that they could get tested for and there are potential therapies that they could get matched to? I mean, that, I think, is a major challenge that we will have to, as a society, as a community of scientists, innovators, and, you know, government and policymakers be invested in. And then the other thing that I think is ripe for coordination is really amongst the companies and those amongst us who develop CDXs. Because, you know, starting from PD-L1 and our own experience with the Claudin 18.2 markers, there are all these different cutoffs and understanding. It takes years to deconvolute whether they all mean the same or they are very distinct. And I think there's a lot of opportunity for collaboration in this space.
I think the big concern is the strategy of biomarker and companion diagnostic going to keep up with the pace of actually us identifying the modalities and the combinations? Yeah. I think that's going to be the real challenge, and unfortunately it seems to be behind, and because it's behind, you know, there's this lag phase of getting these medicines to the right patients.
Yeah.
I think that's a great point. You know, on the biomarker front, you know, I would say at MGB, we're as close to 100% as we can get, yeah. Right? I think it gets complicated as you start thinking about higher order biomarkers, right? Take RAS, for example, right? It's becoming clear it's not just KRAS G12C, but what are the co-mutations, STK11, KEAP1, and then layer in a third biomarker on top of that.
Yes.
So how are you guys thinking about from a clinical development strategy as those biomarkers get increasingly complicated?
Yeah, no, it's a great question, and actually, I mean, a big part of it is that you learn in the clinic, right? That also is one of the realities. I mean, what we've tried to do, one of the benefits of coming in after other companies that have really paved the way in a space is you can learn from that. So KEAP1 is a great point, great example of a mutation that can impact treatment for KRAS molecules. And so we've essentially designed our molecule to address, for example, that kind of a mutation, right? And so you can do that to a point, and you can try to make the best molecule you can. You can understand the mechanisms of resistance as best you can, really understand the on-off in the case of KRAS, and essentially shut down all signaling. But I think at the same time, we're going to be learning in the clinic, right? And so you've got to also have that mindset as you're developing next generation molecules.
I think from a real-world data perspective, there is a lot of, there are companies like Tempus and Caris which are trying to do this. It's, from a drug developer's point of view, it's incredibly important for us to understand the overlap of these biomarkers. So in a space like gastric cancer, it's getting segmented by the different biomarkers. Who are the patients we are treating, and how would they respond to a particular drug depending on what other markers are in place? That is absolutely critical. And we start doing that even as we are thinking about the targets. And in partnership again with institutions like Mass General, trying to understand what that space looks like. A pancreatic patient who has just been diagnosed versus one who has had that first round of chemotherapy, and then some targeted therapy may look very, very different by the time we come in third line.
I mean, I think that's a big challenge in the fact that if you're a patient, you turn up and you have to go through 20 tests to identify which route you're going to be taking. I think that will become a challenge because that's where it's sort of heading.
Well, the lung cancer space is already like that.
It is like that, you're right.
So we have one question from the audience, if you don't mind here. So the question goes, regarding trials and data, have any of the panelists found better ways to diversify trials and data used to develop these novel therapies? Basically trying to get at the point about better diversity equals better therapies. And any thoughts there?
I think, obviously some of the learning is Chris was saying, is you learn in the clinic. And for example, with our ATR inhibitor, we've been looking at a particular biomarker and measuring that biomarker from total loss through to low protein through to high protein has given us a real spread of understanding of how to target it. So a few patients has given us a real clinical idea of really where we should be putting it. So you do learn, and it is important that you do these sort of early experiments as fast as you can from a clinical perspective to be able to then take it to that, take it down the right route, if you want to call it that. If that answers some of that question, I don't know if that's...
Yeah, for us, patient diversity is critically important right at the start. But we alone, I don't think any company alone, whether it's the bigger pharma companies or mid-sized companies like ours, can do it alone. And that's where I think the power of collective real-world data to really begin to understand both geographic as well as racial differences in some of these markers. A lot of research needs to be invested, and I think that's another area of collaboration between, we have data from clinical trials which we can work with institutions like yours to really understand whether certain mutations have an increased prevalence in certain populations. I think it'll help us design the better trials. It'll help us get the, from a drug developing company perspective, if you think of value, we think of, we want to get this drug to the widest possible population. if it's not an effective drug in that population, we would like to know that as quickly as possible and tailor the patient population properly.
But it adds into precision medicine, doesn't it? I mean, that's the whole point, is to be in the right population, the right tumor type at the right time, if you want to call it that. And I mean, it's been used for a long time, precision medicine, but I do really believe we are getting there in terms of better precision in these medicines.
Yeah, but I think it's, the reality is that, we don't understand enough about that topic, right, in terms of diversity, how he maps the biomarkers. And honestly, it's kind of the obligation of, the three of us and other drug, developers and folks to really build that data set, because I think it doesn't really exist.
And I think programs like the Cancer Moonshot and other programs, I mean, they are investing in research that will allow us to get there.
As a clinical trialist, one of the trends I've observed in the last couple of years has been this movement towards not just first line, because that's always a desire, right? But also earlier stage. We're seeing this shift towards precision medicine, even ADCs, earlier stages of disease. Perhaps you could just articulate kind of within your own development platforms, how are you thinking about positioning your agents late stage, early stage in clinical trials?
Yeah, I'm happy to start with that. I mean, I think that certainly you have to be able to address patients that are, like, for example, in the KRAS case, relapse refractory to current KRAS molecules, but also, certainly where you can identify patients that are, haven't yet been on a KRAS inhibitor. And I think that the paradigm is one where ultimately, certainly sooner treatment is better, right? I think in cancer, there's very few cancers where if you don't actually, get diagnosed and understand the genetic makeup early, that it cannot be treated, right? And so, so that is, certainly I think a question. And I guess, maybe back to you, Justin. I mean, at a great center like Mass Gen Brigham, I mean, how do you guys think about identification of patients early and putting them on therapy?
Yeah, I think that things have changed a lot in our clinics, even in the last couple of years, where there was initial hesitation both from patients and providers alike of enrolling an early stage patient because they have by definition curable disease. But I think recognizing that, particularly when it comes to immunotherapies, and as we start getting immunotherapy-based combinations, that those therapies actually may work better when the cancer is still in place, like before surgery. And so that's really opened up this whole new paradigm. And so both patients and clinicians alike have grown to accept that more.
Yeah.
But the way I look at it is, if you can hit that tumor harder and faster earlier, the more chance that you have of that tumor not coming back. And the problem is when you drip feed into a tumor, as a lot of these therapies have been over time, they just adapt. They adapt. And so if you can bring in more of these therapies earlier in combination, so you're hitting harder, then you're actually removing some of the problem that you would create if that tumor can survive and keep growing. So for me, that's why you want to try and shift as many of these new paradigms earlier as possible.
And for us, it's a win-win because these perioperative trials is such an important avenue for us to understand proof of mechanism of our drugs. You get the diagnostic biopsy, you get the surgical samples. The issue and the challenge, though, is that you only get a few doses in, and you have to. Current discussions are whether you need that maintenance phase, whether it adds anything further or not. And I think the correctly designed trials will be required to really address whether that neoadjuvant phase is sufficient or whether you need that continued maintenance therapy. So we are happy to work on that, absolutely.
Well, it looks like we've had a fruitful discussion here, and I appreciate your time.
Thank you so much.
Thank you, John.
Sharing all that you've learned over the years at your companies, and want to thank everyone for participating.
Thank you.
John, thank you.